Okay, so due to its amphoteric property, amino acids exist in various protonated and deprotonated forms. Now, so depending on the pH of the solution, upon which it is dissolved. Note that the fully protonated, or the cationic form, meaning it has a positive charge, exists in the low pH region. While the fully protonated or the anionic form, meaning it has a negative charge, exists in the high pH region. So we'll discuss this in terms of titration on the following slides. We look at your look at your pKa values. Remember we've made mention on the previous lecture about buffers, that when your pH is less than your pKa, your weak acid predominates. Meaning all of your groups should be fully protonated. Whereas when your pH is greater than your pKa, the conjugate base predominates. Now, so ibig sabihin, your groups should be deprotonated. Now, suppose that you have a cationic and at ionic form with charge of the same magnitude. No? So meaning you have your, for example, as shown here, a positive one and a negative one charge. The molecule sandwiched between them is called as your isoelectric ion, wherein you have zero net charge. Remember, remember this because this is an important concept when you're computing for the isoelectric point. So let's ask the question, what would happen if we titrate an amino acid? So this figure shows you the ionization state of your amino acid as a function of pH. Of course, when an amino acid is titrated, the titration curve would represent the reaction of each functional group with hydrogen ions. In other words, the ionization state of your amino acid is altered by a change in pH. Suppose that this amino acid in the given example have a neutral R group. So example, your alanine. The amino and the carboxyl groups are the only titrable groups present. So focus your attention, on the pink portion of this figure. As mentioned earlier, at very low pH, alanine has a protonated and thus uncharged carboxyl group and a positively charged amino group, which is also protonated. As base is being added, the carboxyl group loses its proton, to become a negatively charged carboxylate group, and the pH of the solution increases. So now look at the ionization state. Initially, you have high concentration of the cationic form of your amino acid. As you add base, the concentration of that form decreases because it's being converted to another form. In this case, your zwitter ionic form, because alanine now has no net charge. So what is this? This is your, that is the pKa of your carboxyl group. The boundary. Remember, sabi natin, if pH is less than that of the pKa, then the fully protonated form predominates. As the pH increases further, as you add more base, the protonated amino group, loses its proton. And your amino acid now has a negative net charge of 1. Now look at the green portion or the green line. As your pH increases, the concentration of your zwitter ionic form decreases, while the concentration of your anionic form increases. Again, kasi nga, nako-convert yung zwitter ion since you're losing your proton, and again, and again, nagiging siyang your anionic form. Now this is the pKa of your amino group. So nakita natin na your pKa acts as your boundary. Remember earlier, we've made mention about the isoelectric point. No? So this is the pH. What is isoelectric point, by the way? No? So this is the pH at which a molecule has no net charge. Again, this is the point where sandwich ka ng dalawang pKa provided that your net charge is 0. Okay, so some technique here, no? So for the PI calculation, okay, for acidic amino acid, use the two lowest pKa values. On the other hand, for the basic amino acids, use the two highest pKa values. Okay, note that you can calculate the ratio of each acid to its conjugate base, K, protonated over deprotonated forms as a function of pH using your Henderson-Hasselbalch equation. By the way, the importance of your PI is that you can apply it to your protein purification and characterization technique, no? Specifically under electrophoresis, no? Which we will be discussing on the succeeding lectures. Okay, so now we move to peptide bonds. Okay, so individual amino acids can be linked by forming covalent bonds, and you call that bond peptide bond. Now, it's the special name given to the amide in the amide bond between the alpha carboxyl group, okay, of one amino acid and the alpha amino group of another amino acid. Note that in this condensation process, water is eliminated. Always, always na ganun yung nangyayari. Okay, so your molar mass here of your peptide would be lower by 18 grams per mole. Okay, so your peptide bond can actually undergo resonance stabilization, no? Which makes your peptide bond exhibit partial double bond character, now making it stronger than a normal single bond. Also, okay, your peptide bond is planar, which restricts the rotation about the peptide backbone. By the way, your peptide bond have a trans configuration. Okay, so peptide is the name given to a short polymer of amino acid joined by peptide bonds. Now, they are classified based on the number of amino acid in the chain. So, for example, dipeptide, tripeptide, no? Okay, so these are molecule containing 2 or 3 amino acid respectively. Okay, remember here how you read amino acid. No? You start from the N terminal going to the C terminal. No? So, yung paulit-ulit natin sinasabi, you have your NCC, no? To count the number of amino acid present. Okay, so note that 1 NCC equals 1 amino acid. So let's have an example about this on the following slide. Okay, now, what if naman, if you have a macromolecule containing many amino acid joined by peptide bonds? You call that your polypeptide. Whereas, you call it protein naman if you have, okay, a molecule containing 2 or more polypeptide chains. There. Okay, so this is the example that I was talking about earlier. Okay, so remember, di ba, sabi natin, start ang counting from N terminal going to the C terminal. And that 1 NCC equals 1 amino acid. No? So let's try counting the amino acid present in this example. Okay, so you have here your NCC. Okay, so there, that's 1 amino acid. Again, you have here your NCC. There's another amino acid. Okay, so continue the counting. Okay, you have here again your NCC. So that's another amino acid. Okay, so again NCC. And that's another amino acid. Okay, so by the way, these are your peptide bonds. Okay, so remember, we were able to count 4 amino acid connected by 3 peptide bonds. Now, so we can say that this particular example is a tetrapeptide amino acid or tetrapeptide. Okay. So, yun nga, tetrapeptide. Again, because you have 4 amino acids. Okay, so from now on, we'll be having exercises. So you answer this before proceeding to the next lecture. Now, the answer for this will be included on the succeeding lecture. Okay, so by the way, when you're answering these exercises, make sure that you're not scanning through your notes. Okay, so di kayo best in browsing. No? So answer them based on what you know and what you have learned while you're studying. Okay, so that you can assess how much effort do you still need to exert in studying. Okay, so same thing with your other courses. Okay, so this is number 2. So you can just read this. Okay, this is number 3. Okay. This is number 4. Okay, so this is number 5. Okay, and lastly, this is number 6. So this is the last exercise. Okay, so kindly answer this and try to assess yourselves as I've mentioned earlier. Thank you and see you on the next lecture.